Do you have a class just now? Okay, so no wonder. So you're a bit late. But remember, mark your attendance first. So those who have not marked your attendance, go and mark your attendance first. Okay, got only 11 of you marking your attendance. Okay, so now I cannot actually see your meeting chat. So if there's anything, just talk or someone read to me the meeting chat. Okay, I'm on another computer anyway. So today we're actually going into introduction on Biopython. So the next few lessons is on Biopython, and you'll find that Biopython is much more friendly to use. Okay, so what on earth is Biopython? The main thing about Biopython is allowing us to read different files, okay, especially sequence files and so on. So what are some of the methods? We go through that. So Biopython is actually started, I think, in 2000, okay, whereby a lot of Python programmers, when they want to go into bioinformatics, they realize that they actually need a library, a module, in fact, a package, a large package to deal with all the different components. Okay, so Biopython is actually formed that way. So if you use any other programming language, you use R, there's actually a BioR, bioconductor. If you use Java, there's a BioJava. If you use Perl programming language, there's a BioPerl. So it is used for different programming languages as well. Okay, but BioPython is one of them. It started, yeah, basically in the early 2000s. Okay. So very important, you should have BioPython installed. You can actually go and look at the tutorial as well. So the tutorial actually covers a lot more than what we have. Okay. So this is a Biopython tutorial book. Okay, covers a lot more, and it's pretty well written. Give you a lot of information as well. Okay, you can deal with, let's say, sequences. You can deal with annotations. We'll talk about input output. You can deal with multiple sequence alignments. You can actually run BLAST on it. How do you access the NCBI databases? Everything is actually listed here. Okay. So even does with a lot of structural biology as well. Okay, how do you compose atoms and so on and so forth. Okay, population genetics, phylogenetics as well. So all these you can use it. Although we do not, most of the time we seldom use everything. Okay, so that can be quite useful. What you can do with it. So do take a look at as well. Okay, if you need help, then you go to Biopython website. Okay, it actually gives you a lot more information. You can set up the mailing list as well. Okay, there's tutorials, there's some examples and so on. But the tutorial, the cookbook, which is essentially this is a tutorial and cookbook. It gives you almost all the information you need. Okay, so far so good. So this is Biopython. So we'll actually cover a little bit of it today. So Biopython package, the main thing it does is it contains parsers. Parsers are essentially file readers. So how do you handle FASTA format? How do you handle GenBank format? And so on and so forth. So they can be actually quite useful. After you handle all these formats, can you access online databases? Like that when you want to search, okay, let's say you go to NCBI. Okay, this is called Entris. Okay, Entris, ENTRIEZ. Okay, this Entris search engine. Biopython can actually access all of these as well. So that makes it pretty useful in that sense. And common interfaces, like for example, how do you run BLAST? How do you run cluster W? And so on. So in your sequence analysis project, you can actually use Biopython to do it if you know how to do it. Okay. Otherwise, I'm sure your project currently you use Mega, right? You use Mega. So that is one tool that you can consider. Okay. So to start off with, make sure all of you can import Biopython library. So the first thing is to check whether can you import bio. Okay, if there's no error, at least it should give you the correct stuff already. That means Biopython is installed. So why not you try it out and see whether does you install Biopython or have you installed that? Let's go to second place to see whether is there a better way to do this. Okay. So anybody have issues with, that means you have not got Biopython installed. Okay. Let me see. Teams. Okay. So after that, you can actually look at the version number. So you type this bio _, version. Okay. You should have about version 7, 1.7. Do all of you get that? Hey, response day. Okay, motor is all right. Okay, so if you have this output, that means your Biopython is installed. Okay, if you don't have it, then we have to deal with it. So I presume all of you have it. Okay. Okay. So the next step is how do we run sequence files? Okay, so a lot of our file format, have you learned the different file formats? For example, your two file format, your FASTA and your GenBank. Have you learned the differences between these two formats? Okay, I'm sure we've covered it in front already. How many of you still do not know the difference between these two formats? Anyone? If you're okay, say that you're okay. You know the difference. Okay. Okay, the main thing is how do we read these files? So when we use Biopython, it makes your life easier. We tried to read your FASTA file in the last tutorial, and you find that it's actually not that easy. Okay, so in this case, we actually use this bio.sequence.io library. So this is a sequence input output library, and we'll be using that. Okay. So let's go to a FASTA file. Let's go to a FASTA format. I will go to my nucleotide. Nucleotide, where am I? PPP nucleotide. Okay. And I want to find a protein called, I will have it there, Sonic Hedgehog. Sonic Hedgehog is a protein or it's a gene. And I pick the first one, let's say Sonic Hedgehog gene, Homo sapiens, complete CDS. So I click on this. So this format you see is actually called GenBank format. It consists a lot of information and it's on the DNA. So we may not want DNA. Let's see, do we have a bit more closer example? Okay. Scalars, we know. Okay, let's look at this variant 1. So it's mRNA, variant 1. Okay, so this is called a GenBank format. It contains a lot of information for you. Of course, you have a sequence as well. Okay. The few things that you have to know on GenBank format, which is quite useful, is the accession number. There's this difference between accession number and version number. okay? Normally when we write, we have the whole version as the accession number. So this is the accession number as well. But in reality, the last digit, 0.4, means that it's the fourth update. Okay, the accession number is this, but normally you have it in the URL as well. So the accession number is very important. You can go to the different versions of it. Let's say the first version or the third version. You can go to 0.3, which is the third version. Then it will tell you that this sequence is updated and the date is 2000. This sequence is 2018. Okay, but for version 4, the date is 2020. So it's just a few days ago. Okay, so far any problem? So make sure you, when you get the sequence file, make sure you have the correct version number as well. Okay, don't get the wrong version. All right. Another aspect that is quite important is this thing called organism. This is the whole lineage of Homo sapiens. So yeah, eukaryotes. Anybody know what chordata means? Let's say if I click to Homo sapiens, you go to a taxonomy browser. So this is the taxonomy of human. Okay, so yeah, eukaryotes. Then you come to, you are bilateral. That means you have a left and right. You are under deuterosomia. Anybody know what does deuterosomia means? Yes, no? No? Okay, deuterosomia simply means that in the early part of your development, you are literally just one hole. You are just one tube. Yeah, just a hollow tube. That's how human life is started. You are just a hollow tube. Deutero means two. So you are actually, it's a tube made out of two holes. Okay, so effectively, the two holes is now your mouth and your ass. Yep. When you are just when you are about 48 cell stage in your ovum, 48 cell stage, that's your blastula. Your mouth and your ass come from the same place. Okay. So chordata. Anybody know what chordata means? Okay. Chordata simply means that you have a spinal central nervous system, a spinal cord. Okay. Crania, you have a skull. Vertebra, you have a vertebrate, you have a backbone, and then goes on down. Tetrapodia tells you that you have four limbs. Amniota tells you that you have amniotic fluid sometime in your developmental process. Mammalia tells you that you're a mammal. And then it comes to a subclass called Eutheria. So mammals, there's three different types. Eutherians are those that have placenta. Monotremes are those that lay eggs. Marsupials are those that with a pouch. Okay. Then go down, we have primates and so on. Okay, so it gives us a long taxonomy. Currently, we actually consider Neanderthals and Denisova are actually part of Homo sapiens species. Okay. So how do you read this file? If I give you this file without using Biopython, it's quite difficult to read. You have to do the parsing yourself. You go to the FASTA format and then gives you the shortened FASTA format, which you have seen before. So your description line and so on. Okay. Now, we'll be using that for a while. So let's say you are given a FASTA file. Okay. In this case, the FASTA file that we are giving you, or I made for you, is inside your tutorial, your Sonic Hedgehog. Okay. So Sonic Hedgehog. So where on earth is my Sonic Hedgehog? Okay, Sonic Hedgehog. Let me see if I open it here. So Sonic Hedgehog is a FASTA file. I have multiple sequences. I think 8 or 10 sequences. So how do I read them? Okay, so this is the description line, which is a bit complicated. Then this is the sequence that you have. Okay. So now, what we want is, how do you read a FASTA file? You actually, first, you need to import the sequence IO. So that's the first step you have. You go there. So let's go to our Python console, and then you make sure that you can import sequence IO. Okay. After that, you actually use a file reader. So this file reader is almost this sequence IO.parse. It is very similar to what we have done your open function. It actually uses open function. So let's take this and see what happens. So what I did is that it passed what file. So you need the whole listing, the whole file path. Then what is the type of file that you're giving? You're giving a FASTA file, then put FASTA inside. Okay. So now I have a set of records. For every sequence record, I print out the ID, which is the string, the description line, and I print out some of the sequence and then the length of the sequence. Understand? So let us just take this and see what happens. Okay. Make sure it's double slash. Single slash will not work. Okay. And so after that, you just produce this listing. So you have your ID line and so on. Remember, the ID is just half of it. Okay, it just basically up to here, up to the first space. Okay, the rest of it will not be included. So far, so good. So it prints there, and then it prints a bit of the sequence for you. REPR stands for representation. So it's a printable representation. The length of the sequence record, this is the sequence itself. How many bases it has. Okay, so far, all right. So basically, this is how we read FASTA file. Okay, if we want to read the sequence itself, let's say we want all of the sequence. So I don't put the representation. I can also do that. It will print out all the sequence for me. Okay. So now it prints out the entire sequence, which is exactly what I have inside my FASTA file. So far, so good. Any problem up to here? Any issue? Hello? No, Jasmine, you're okay. The rest? Joanne, Erin, Ryan, you're fine. Lionel, you're okay. Junhang, fine. Nabil, Yun Hui, Dong Te, Hazel, okay. Where is Rebecca? Chloe? Yep, Chloe is fine. Rebecca and Johnson. Are both of you in? Johnson, you're okay. Rebecca? Has Rebecca sunk? Rebecca, are you alive? Okay, good. All right. So let's take one piece of data and see what happens. So let's say we call it a record. Let's say we take the last record to actually see what is happening inside. Okay. So we want to see what is actually inside record. Record has a lot of other things. So you see, the ID is only the first part of the whole description, but all the information, the entire line, this is called the whole descriptor line. This thing is called the description. It's actually stored under this, the description itself. Okay. So all the information is still retained. You can process the description if you want to. So record dot description. You'll print you the description itself. Then you can process the description if you want to. Okay. So for example, the description, I want to split it according to the spaces. Okay. So I can put description, then I do a split. I split whenever there's a space, I split it, but this is actually very rough way of doing things. Okay. So you'll find that it looks something like that. The Sonic and Hedgehog is kind of separated already. Yeah. So it's a bit rough in that sense. Okay, it's not easy. Okay, but the first part, this is actually your ID. The ID is usually also the name as well. So far, all right? So at the very least, it helps you to read your FASTA file. Now, although reading a FASTA file is not a very big deal, you have read FASTA file from scratch last week. But when you come to your GenBank file, then it's a big deal because GenBank file is so hard to process, so difficult to process. Okay, so let's look at that. Let's look at GenBank file in the first place. So the FASTA file, so we don't bother with the FASTA file for now. Let's look at how do you process GenBank file. The same GenBank file, I have it inside Sonic Hedgehog.gb, the GenBank file. So this is the GenBank file for all the few Sonic Hedgehog genes I have. Okay. How do you read GenBank file is also very easy. All you need to do is the pass, this is a file name, give it a GenBank file. Oh, GenBank file, this is my GenBank file. And the file type, instead of FASTA, I change to GenBank. GenBank is GEN, not GNE. Okay. Then you will read the GenBank file already. Okay. So now you're actually reading a GenBank file, you just run it, and you're reading a GenBank file now. Okay. So let us see the record, the last result. So this is actually your GenBank file. See the description, the ID is different now. The ID inside the last one, right? So you see, this is a locus. This is a session number. Okay, so you go there. This is the accession number. This is the locus. See these two. This one is the locus. One is the accession number. So it maps to the ID and the name. Then the description is actually just this line, the definition. Okay. All right. Makes sense, right? All of you, can you see what's happening? So reading GenBank file is easier this way. Any questions so far? No? Okay, so my question to you is, does that mean that, you see, one GenBank file record has so much information? Does it mean that all the rest of the information like references, organism is being thrown away? All these features are being thrown away. Do you think all this information is actually being thrown away? No, right? Unlikely that it's being thrown away. So let us look. We can go to use dir to see records. What is actually inside? It contains a lot of information that we can have. Okay? So for example, we can look at annotations. Okay, let's do annotations. Record annotations. See, this gives you a fair bit of annotation. Tells you the molecular type, topology, the date, file division, date, accession number, source, organism, the taxonomy itself, the reference itself. This is easily equivalent to almost what you have from this section here. See, this is a date, everything. It actually gives you all the information up here. It is just embedded somewhere, the annotations. Make sense? Okay. Then you can even go into the references. So the annotations, you want to go into the references. Okay, it doesn't have it. Dictionary doesn't have the references. What does this produce? Put references. Let's try this. Okay. You see, now you can produce the reference itself. And the reference is actually another data type. Right. So you can go dig down, what on earth is it? Okay, the reference, the title, and so on. So does it have things like title and all this stuff? So I can go to references. I can even go to reference. Let's see, not sure. Let us try. Let's dig a little bit inside. Let's go to title. Okay, so references. For example, I have the first reference, title. First reference. Yeah, it gives us a reference. And then here, this is one reference record by itself. So you can continue digging a bit deeper. So this is a reference record. Okay. Inside a reference record, you can have your journal location and title. So this, then I have journal. Tell me which journal. Oops. So let's say for this reference number 1, which is index 0, I can go and find the journal. Okay. So this is a journal. Then what is the title? So this is the title of the journal, of the article. I can even go and find look for it, the authors. Does it call authors or authors? I have to hunt it down. Authors. Okay. This is my author list. that is essentially under this reference. So this is an author, gives me the same set of information. I also have the second one. Let's say, how about for reference number, the second reference? I also have it. And I can have the title as well and all this. So that is the information for this second reference. Okay, it's a direct submission. So all of them can be listed. Then remember, we have these features, is even more complicated. Okay, you can start to dig in all the individual information inside your gene bank. Okay. So it is a very complicated set of tools for you to do collect all the data that you want. Now imagine if without using Biopython, how on earth are you going to get all this information in a correct format? It will actually take you a long time. Okay. This is actually why Biopython is developed in the first place, is to resolve all these problems with reading different files. So far, any issue? It takes a lot of exploration. Please go and explore it yourself. Okay. Like for example, I can go into record. So features. Okay. See, record features. I have feature number 1, feature number 2, feature number 3. So this is the source, gene, and CDS. So it corresponds to this feature number 1, feature number 2, and feature number 3. So I'm quite sure that all the information are inside. Okay. So if I go to feature number 3, which is feature 2, then I find out how do I actually read more stuff inside. I go to dir. See, it contains a lot of different stuff as well. Okay. Translation and all these things. Okay. I can have location. So it will correspond to the location here and so on. So I can have the last feature and want to find out the location of this feature. So I type location. So it tells me the start and stop position. Okay, and then this strand 1. Okay. So please go and dig through how to use Biopython, especially with the gene bank format. Okay, we'll look at that in more detail in the subsequent lectures as well. But if you have time, please go and dig through it. So here, essentially, this is how I pass the gene bank file. I've shown you already. I just change the file name to a gene bank file, and my type, I just change it to gene bank file. So this is the easiest. Just these two items. Oops. Okay. All I need is to change the file name, give me a gene bank file, and now what the file type is change it to gene bank. Okay. That is the easiest way for us to read the gene bank file in the first place. How you want to deal with it, we talk about it elsewhere. Okay, so this is the easiest two ways of reading your FASTA file and gene bank file. Okay. Next, we come to this thing known as sequence objects. Okay, so sequence objects is actually also in subsequent lectures. Let me show you. So the next lecture is on sequence annotation, objects, and so on. But we'll cover a little bit of it here. So your sequence, actually, the sequence in your FASTA file or your gene bank file is actually held under this sequence object. And because it's held under sequence objects, Biopython also has convenient tools like how do you do translate, how do you do reverse complement. Remember last week, you have a hard time of how do you do reverse complement and so on. This makes it easy. So what we did last week for tutorial is for you to know how difficult it is if you were to do by hand. Okay. And what type of alphabets it is. So you go through a bit of that. The most important is this thing called alphabet. There is a set of alphabets that's actually predefined. It uses this IUPAC alphabet codes. That means your DNA, RNA, and peptide codes. Okay, so these are important because it allows us to do what we want to do. So the normal protein UPAC, IUPAC contains your basic protein class, your 20 amino acids. Okay. Then we have these extended amino acids. For example, we have selenocysteine. Okay. Pyroxene, pyrrolysine. So these are additional amino acids that your body can use. Okay. Sometimes we have ambiguous symbols like B, which stands for either asparagine or aspartic acid. Okay, glutamine or glutamic acid. Okay. Sometimes, so X, J stands for leucine or isoleucine. Isoleucine. X means some unknown amino acids. Anybody know what is another name that we tend to use for aspartic acid? Remember when we do a biochemistry, what is another name that we tend to use for aspartic acid? Sometimes I use that as well without even knowing. Anybody remember? Or glutamine. What do we always call glutamic acid as? Glutamine is not glutamic acid. Glutamine is glutamine. Glutamic acid is glutamic acid. Everyone knows what is another name for glutamic acid? Hey, sleeping or not? No? Anyone remember? Ambrose, do you remember? Glutamate, correct. Okay. So glutamic acid, sometimes we also call it as glutamate. Okay. So when biochemists call glutamate, it actually means glutamic acid. Okay. So aspartic acid is also known as aspartate. Okay, so all this you have to be a bit more practice. Okay. Your DNA itself, you have your 4 basic nucleotides. You can also provide ambiguity. Okay. For example, we can look at some of it. Okay. Code for nucleic acid. So you have the 4 basic types that you know this, your A, T, C, G, but we have other more convenient types. For example, R can mean your A or T, which is, I think, pyrene or pyrimidine, can't remember. One is pyrene, one is pyrimidine. So your S means there is 3 hydrogen bonds, W is 2 hydrogen bonds, and so on and so forth. Okay. N means every single base. So you have extended code as well. Okay. So this can be very important, which means that actually you can design your primers with non-specific nucleotides. Okay, so please be careful. Not all the things will go wrong. sometimes we want non-specific nucleotides to make sure our primers is actually more flexible. Okay. Same thing with RNA as well. So let us go, how do you actually build a sequence? To build a sequence, you have to import the object sequence from sequence biosequence. So let's do that. So you have to import first. Okay. Then you just build your sequence by using the object. So sequence is object. Then you put in whatever sequence you want. Okay. So the basic default is that just a alphabet. That's the basic default. Okay, it's just an alphabet. For Python Biopython 1.7 onwards, it doesn't print out the alphabet. Okay. You can convert it into a string if you want to. So to convert into a string, because it's an object, you can convert a string by just typecasting it using a string. Okay. Gets back to a string for you. But remember, because this is an object, it contains a lot of other functions. So I can see what I can do with it. Okay. So I can do complement. I can do reverse complement. You can transcribe it, you can translate it. So let's say this is what we have, right? My sequence. Okay. If I do the complement, why do I always do this? So you see, the complement is as this is what we did last week. You do the complement of it, but this is from 5' to 3'. This is from 3' to 5'. Okay. So this is 5' to 3'. But when you do a complement, it will give you just a complement, but it's from 3' to 5'. Most of the time, you want to reverse complement. Okay. So we do a reverse complement. And then this gives me, this just reversing this lah. So it gives me a 5' to 3' again. Okay, so exactly this is what we tried to do last week. Any problem so far? Do you prefer to use Biopython, or do you want to do the same what we did last week? Which one do you prefer now? Biopython, right? Yeah, because all this is tested properly for you. Okay. Then you have this sequence. Assuming that this is your DNA, I can even do your transcribe. If this 37 is DNA, right, I transcribe. It will give me the corresponding RNA for it. So it's just basically changing the T into U. And then it gives us the RNA alphabet. Make sense so far? Can you see what's happening? This is the DNA. This is the RNA. Okay. I can do even more. I can even do a translate. Okay. Translate, basically, it translates. This is one amino acid. This is another amino acid. AGT is one amino acid. CAC is one amino acid, and then this is the third amino acid. So it gives me SHS, which is serine, histidine, serine. But it gives you a warning. It tells you that the sequence you give is not a multiple of 3. So which means that the last A is actually not used. So it gives you a warning, that's all. So far so good? Okay. Okay, so essentially that is what the sequence object does. Let's say we give it another object. We go to alphabet and import this IUPAC codes. Then we can use it. Okay. So now we have a sequence, and we can call this as an unambiguous DNA because the code is actually unambiguous DNA. Then now my sequence is your unambiguous DNA data type, sequence type. So this actually helps us a lot if you can actually define that for yourself. Okay. Then let's say when we have a sequence already, we can go to Biopython sequence utils. Okay. So let's say if we want to find out what is in sequence utils, we can go to import let me think, what is the easy way? In fact, we can go to Google and find out what is in this sequence utilities. Biopython, what is in sequence utils. Okay, so when we go and read the API, for example, when we read the description, the documentation, what you can do, you can calculate the GC content, you can calculate the skew, you can calculate the molecular weight, and so on. So this is the very first part. You can calculate the isoelectric point. Okay, so let us do a bit of things at one go. For example, when we want to just calculate the GC content. Okay, remember we have been calculating GC content for a while already. We just import it. And then you calculate the GC content of your sequence. Okay, so it gives you about 45.5% GC content. You can take this and then you calculate your own GC content. That makes your life a lot easier. Okay, so you can actually do a lot of analysis this way. Okay, so let us just look at a few other things. There's a little bit off. For example, you want to find the molecular weight. Okay, very often you want to find the molecular weight. So we import GC. Let me just look at the description first. Okay. So let's say if I take here and just import everything, make my life a bit easier, import everything, which I don't really quite like to do, but assuming I do it. Okay, then I just see what things have I imported so far. Probably a lot. Okay, I can import the molecular weight. Okay, so I'll just say you probably can't see my screen now. Molecular weight, and then my sequence. So this gives you the molecular weight. But do you know what is the unit here? What is the unit for this molecular weight? Anyone make a guess? Daltons, correct. So it's in AMU, atomic mass unit. Okay. You do know that one atomic mass unit equals to 1 Dalton. Okay, so you know that in your secondary school already. So effectively, my sequence here, this short sequence is effectively 3.4 kilodaltons. All right. So far, so good. So let's try something. Can we just use this and randomly just throw in some awkward sequence? Maybe that will work. Not sure. You will calculate for me as well. Okay. So this is a useful way to calculate any sequence that you want. Okay, so this is not for DNA, so it cannot, it doesn't recognize. But you can change the sequence type into protein. Then you will actually calculate the protein sequence for you, molecular weight for the protein, for a peptide. Okay. So far, any problem? Let us just see a few other stuff, can be quite useful. So you should know what on earth is isoelectric point, right? Anyone know what is isoelectric point? Anyone know, remember what isoelectric point means? Hazel, remember what isoelectric point means? Okay, what happens at isoelectric point? Where the protein, pH where a protein is neutral charge, correct. So if a protein, you put it, you solubilize it in its own isoelectric point, it technically the peptide will not dissolve. It will actually precipitate out of solution because the charge is 0. Okay, so that can be very useful. We can also use it to determine, let's say, melting point of your nucleotide sequence. So this itself, if you calculate the melting point, it will be very similar to calculate. Remember how you calculate the melting point for your what do you call that? Primers. Okay. But actually, there's a lot more to calculate melting point. For example, how do you do a salt correction? There are different equations for it. Okay. The Wallace rule is the easiest that you learn. Your 4AT, 4GC plus 2AT. Okay. You can actually change it based on the salt calculation as well. Okay. So it can be a bit more tricky. Okay. The salt calculation, this is actually the equation for your salt calculation. Okay. You can actually calculate, change it based on your chemical content. For example, your formamide, DMSO concentration actually makes a difference. Another one that's quite useful is your protein parameters, simple protein parameters. You can go and look at it. Okay, you can calculate molecular weight, aromaticity, isoelectric point, second molar extinctions. Do you know how to use molar extinction? Okay, aromaticity, stability, and so on. Anyone know how to use any of all this? Okay, so let me show you. This should be shown in your structural biology, but let me show you how it looks like. Let me get another sequence. Let me see. Hopefully I can find this sequence. RSEC8, nucleotide complex. I'm RNA. Yes, okay. So I go to get the protein for this. This is a yeah, anyway. So this is a protein sequence. And I just go and find, let's see, it's true. Okay. You haven't seen these two before, you can ask your structural bioinformatics lecturer, I think Mr. Paul, to show you. So let's say if I have a sequence, this is my mRNA, this is my peptide sequence. Okay, so it's a endocyst complex. So let me just take this sequence and throw it in. Does it actually copy and paste amino acid sequence? Then I compute the parameters. What does it give me? Okay, it gives you your amino acid concentration, composition, the molecular weight. So this protein is 110 kilodaltons. The theoretical isoelectric point is about 6. So based on this, you should be able to figure out how to purify this protein. Okay, carbon composite. The formula is ridiculous. Don't bother about this. Now, extinction coefficient. Anybody remember this? What does it mean? Look at the description for extinction coefficient. I know it takes a little bit of reading. What do you think it means? Anyone? Can make a guess. How do you use this number? 70095. Anyone? Okay. Light absorbance. Okay. So when you remember when you do your spectrophotometry, why on earth do I have this? Okay. When you do your spectrophotometry, your cuvette. So this is a light source, and then this is a detector. All right. So remember your equation is your absorbance equals to what? So remember the equation? Ryan, still remember the equation? Or anybody can help? So this is your sample. Jasmine, do you remember? Do you do analytical chemistry? Log something OD 280, 260. Yes, okay, so this you have to set to 280 nanometer wavelength. If this is a protein, protein. Okay. Uh, Not exactly. Okay, I'll go to that. So the equation is epsilon, concentration, length. Okay, this epsilon is your extinction coefficient. Okay, so this is your concentration. L is your path length. Path length simply means what is the length here. Your cuvette is standard size. Your cuvette is always 1 cm. So path length is always equals to 1, which means that your absorbance equals to epsilon times concentration because path length is 1. Make sense? So what it really means is for this specific protein, the extinction coefficient is 90079 70095. Okay, so here it is actually absorbance equals 70095 times concentration. Okay, and if the concentration is 1%, 1%, that means 1 gram per liter, absorbance will be equals to 1. Okay, so you can take the sample and just go and measure your spectral. You don't have to do your Komazi blue staining and all this stuff. Okay, for Rebecca, your OD is not it's not 360, it's OD 260 divided by OD 280. This has to be between 1.6 to 2 for pure DNA. Because OD 260 measures DNA. Okay. Wait, if you ever work, if you in future work in the lab, you'll end up with another thing called OD 260 divided by OD 230. Okay, this is also important, just that you seldom use it in your class. But I have no idea why it was never mentioned to you before. OD 230 is to measure your phenol. A lot of our DNA extraction kits contains your phenol, so this is to measure phenol contamination. All right. So essentially that's it. Extinction coefficient, the stability, and so on. So it's a very good way for us to look at it. Okay. So this especially also contains a lot of other things. I can go through a whole day just talking about it, but I will not. Okay. Now, because DNA sequence, the sequence objects, you can actually add them up, concatenate it. Okay. So let's say I can actually have two sequences. So I can do this. I can have a sequence ATGC. I can actually concatenate it with another sequence. It may not make sense at all, but you can produce them together. You can, because they are considered just alphabets. You can even concatenate a DNA sequence with a protein sequence. Although it doesn't make sense, it will still join for you. Okay. 53 still makes sense because both of them are nucleotides. 54 doesn't make sense because one is nucleotide, one is amino acid or peptide. But it will still join for you. So far, okay? Okay. Now, so far, what you know is it doesn't have a join method, so you have to use concatenation. Okay. Why not you do this yourself? Since we can join everything, but if the sequence, you actually give it a specific name, a very specific type, it will give you an error because you cannot join protein with DNA. Okay. But just now, why did we join protein with DNA? Because we never give it the type option behind. So if we give this a type option, okay, so let's say we call this as your protein and the other one is your DNA. So this is your unambiguous DNA, but this is your protein. Is it? Can I get a spelling right? Protein. Okay. Then you cannot join because by right you're not supposed to join DNA to protein. 54 just treat them as though they are just alphabets, but here you actually 55 define the alphabets as DNA or protein. Then you cannot join it. Okay, so that is an important check. So far, any problem? Do you understand the difference between this 54 and um, 54 and 55? Why one work, one doesn't work? Why 54 works and 55 doesn't work? Or those who get it, just say that you're fine with it or you're okay. Why 54 works and 55 doesn't work? The Ambrose is all right. The rest, just Ken, Ryan. Hazel, all right. Yun Hui, okay. So almost all. Nabil, have you responded? No, lost. Nabil. Okay. Now, what happened to 56? Did you compare 55 and 56? The same sequence, right? But why I can change this into protein? Anybody can give an explanation what on earth happened in 56? I now want to give you a go. What happened in 956? Why does it work now? Okay, I set the DNA label as protein. And it happens that ATGC are also amino acid codes. Okay. It happens that ATGC are also amino acid codes. Now, this is a very important thing. Okay. Let's say if I were to go back here, Let's say I take the mRNA. Okay. Now this should not work already. In the past, it used to work. So I have a mRNA sequence here. And remember, mRNA is just ATGC. Okay? So I put it inside this protein parameter calculator. And I compute. At the moment, it gives me a warning, I think. No, it doesn't even give me a warning even. In the past, okay. So you see, it will still calculate for me. So some tools will give you a warning. This tool doesn't even give you a warning. So it assumes that your protein has only 4 different types of amino acids. And it will calculate, again, your isoelectric point, which doesn't make sense because you are actually using a RNA sequence. You haven't translated it into a protein sequence yet. But the program doesn't care. So please take note, a lot of program doesn't care. Some program will give you a warning saying that it doesn't look right because you have only 4 amino acids, which is a bit strange. But actually, that's not a wrong answer. Okay. So here gives you a 288 kilodalton. So it's a super big protein. Okay. You always have to check whether the sequence given to you, is it a gene? That means your DNA sequence, is it an mRNA sequence, or is it a peptide sequence? Because if the gene given, if it is given to you as a DNA sequence, a gene sequence, it will even be worse. Because gene sequence in eukaryotes can have your introns. All these are the mRNA. So if it's a gene sequence, it can have your introns. So please be careful with it. Don't just take the whole sequence and process it without even knowing what on earth it is. I've seen this mistake far too many times. Okay, so far so good. Okay, now if you have two different alphabets but they're compatible, for example, generic nucleotide tells you a nucleotide and unambiguous DNA, they are compatible, you can actually join them together. No problem with that. Okay, they must be compatible, must be logical for you to join. So sequence objects, we provided a few, your complement, reverse complement, transcribe, and translate. We have seen that before, and it can be very useful. Okay. So let's say we have this, we go back. I've shown you just now, so let's do it again. So let's say this. Okay, or else let's pull out one thing from our previous tutorials. Tutorial 3. Let's use back our MET kinase again. Okay. So this is our sequence, our dearest MET kinase sequence, which we love so much. Okay. And then now I have put these codes in. And I just call this as my sequence again. Sequence equals to sequence. I know this is a bit confusing. Okay, just say. So this is your mRNA. And then this is your DNA, unambiguous DNA. Okay, so far so good. Can you see this line? So if I run it, we have to save first. Okay, so let's don't bother. We just use a copy and paste here. So now I have my mRNA. Okay. mRNA is a unambiguous nucleotide, unambiguous DNA. Okay. Then from then, I can actually do a few other things. I can actually do, let's say, mRNA. I can do a complement. Give me the complementary sequence. I can do the reverse complement, or I can do the translate. So this is the complement AA. Okay. So there's an error here. Okay, that means the last AA is actually not used. Yeah, the last A is actually not used. So you have to kind of remove that. So let us go to here. If you take this, you can do the complement, reverse complement. Okay, but what you important is you cannot do the complement of a protein sequence because protein doesn't have a complement. Okay, protein sequence doesn't have a complement. It will give you an error. Transcription, you should be able to do that. So transcription, you transcribe, give you a messenger RNA, that should not be a problem. We have shown you a few times already. Or you can do it based on what we did last week. Okay, but Rebecca will definitely prefer the current method than what we have last week. Okay. So again, this is where you have to test your sequence itself. Let's say this guy, your coding sequence. Okay. Then you can do your coding, DNA, translate, and make sure that you get, oops. So this is your translated sequence. So this is one amino acid, this is another amino acid, this is the third amino acid, and this is the fourth amino acid. Okay, so you have only 4 amino acids. So far, any problem? Okay. So from the RNA to DNA, you can do your reverse transcribe. Okay, it's almost like doing your reverse transcription, but it's not called reverse transcribe, it's called back transcribe. So back transcribe is actually same thing as your reverse transcription. All right, so far? This is actually your reverse transcription. Any problem? Okay, so translation, it works the same way, with the exception of a few things. What happens if there are stop codons? Okay, so let's say for this sequence, for example, let's say this, we call it mRNA. This sequence. Okay. So if we transcribe or translate what it means is this is a stop codon. Asterisk is a stop codon. So it tells us that AUG, so this is A, up to here is, so this M, this A, I, so this is V, this is M again, methionine again, this is guanine, this is R, then this UGA is actually a stop codon. Then it continues translating for you. So you just print out a stop codon and continue translating. It doesn't stop at the stop codon. Make sense? So just directly translate, it doesn't give you until the first stop codon. So far, so good. So UGA is a stop codon, which is this. Then the last one, this is also UAG is also a stop codon. So far, any problem? Okay. Remember is translate function does not stop at the first stop codon. Okay, this is important. Because otherwise you take the protein sequence, it will give you totally way off something crazy. Okay, up to here, any problem? If you're okay, then say that you're okay. All right. So and okay. All right. Okay. So more or less, yeah, quite all right. So let's continue. Just have a little bit more. Yep. So it'll give you a stop codon. That's one thing. Now, sometimes if you want the when you hit the first stop codon to stop, you actually have to give it another option called to stop equals true. So let's go back. And you will give it an option to stop equals true, right? It will then stop at the first stop codon. Okay? Natively, it doesn't. So natively is as good as true to stop equals to false. Now this stop at the first stop codon now. Okay, so the options, please remember. All right. Okay, now the next important part, I'm not sure how many have you even covered this before. It is that the the genetic code that you use is called a standard genetic code, but that is not the only genetic code available. Okay, so for example, I can show you this. Okay, so I have the sequence is one, right? I show you this. I say that now this is I say table one. Table one will give you this. Table 2 will give you this. Table 3 will give you this, or something different, or maybe be different. See? Table 1, Table 2, Table 16 are different. So what on earth are these tables? Table 1 is your standard genetic code. Table 2 is vertebrate mitochondrial genetic code, which technically means that your mitochondrial gene is translated differently than your genomic genes. Does it make sense? So genetic code is not universal. To some extent, it's universal to the extent that 3 nucleotides give you 1 amino acid. So the codon to amino acid still remains, but the translation may not be. Okay, this is very important, especially if you are like me, tends to work with a few other weird species. Then you can go and look at this information about different translation tables. Okay, let's go and look at it. So keep this handy. Look at this. There's actually a whole list of genetic codes. Okay, the standard code is the one that you learn in your first year when you're doing your genetics, molecular biology, all this. But we have this thing known as the vertebrate mitochondrial code. Then the yeast mitochondrial code is different again, and so on. Okay, you have your bacterial plastic code. So bacterial, archaeal, and plant plastic code, this is used for your chloroplast. So mitochondria is very weird. You have a few different other codes. So most of them are, and then you have some of it, the nuclear code is even different. So what are the difference? This is how you read. Okay, do you know how to read this table? Okay. So how do you read this? So standard genetic code, right? So TTT, first base, second base, third base, TTT give you a F. So let's check methionine. ATG give you a methionine, and so on. Okay. The asterisk is the stop codon. Initiation codon is AUG, but then there are other alternative start codons as well. So ATG is not the only start codon. All right, ATG is not the only start codon. Now, vertebrate, you also read the same way. Then here tells you what is the difference between code 1 and code 2. For the standard genetic code, AGA is arginine, but your vertebrate mitochondrial code, AGA is termination codon. Okay, AUA is isoleucine, but in code 2 is methionine. So it tells you the differences. Then there are alternative start codons as well. For example, BOS. Any know what are BOS? What does BOS means? What animal is that? Cow. Yes, cow. Good. Okay. Yeah, so you have a lot of different codes as well. Some is just one difference and so on. Some can be a few other differences. Okay. So with that, let me just go through one other thing. By now, you should know common animals. Let's say find it. Okay. Okay. Where am I? Okay. So let's look at common names. Okay. So you know that Homo sapiens is human, hopefully. Cow is Bos taurus. Okay. So mouse, do you know the Latin name for mouse? Anyone know the Latin name for mouse? Mus. Mus is the genus name. Mus musculus. That's mouse. Okay. Let's see. What about the lab rat? Let's find protein. Let's say insulin. Maybe this will be easier. Ratus navaticus, correct. But Rebecca, make sure that your N is not capital. Around there is nothing very wrong. This. Okay. You can see here. Okay. They know Rio. Anyone know what it is? They know Rio. This guy. Some kind of bird. No, it's zebrafish. They know Rio is zebrafish. Okay, it's zebrafish. Okay, they know Rio is zebrafish, not a bird. Mus musculus is your mouse. Ratus norvegicus, that is your Norwegian rat. This guy, hopefully you know. Anybody want to try? What animal is this? Which guy? No. Samo salsa. Where my cursor is. Salmon, correct? Yeah, that's salmon. This one you probably do not know. Screw-scopha. Pig. Yeah, Screw-scopha is pig. Okay. Bos taurus, Gallus. Chicken. Okay. Drosophila, you know. Canis lupus. No, Canis lupus is not cat. Wolf. Yeah. So what is the difference between wolf and dog? Domestic. Okay, domestic, fine. So dog is Canis lupus familiaris, means familiar dog, familiar wolf. Okay. Ventriculus, this is actually a chimpanzee. Nothing to do with the personality. This is another monkey, macaca. Okay. So Canis lupus familiaris, this is your dog. So a dog is actually just a familiar wolf. For Ambrose, it's a cat. Which cartoon character? What do you think about cartoon characters? What does cat always associate with in cartoon? Big dog? Oh no, nothing to do with dog. Felix cactus. That's your regular cat. Felis cactus. Okay. So you have to get familiar with a few of these names. Similarly, the next one I want to get you quite familiar is, for example, when we use this word, okay, murine. What animal are we talking about? Which animal when we call murine? Let's say we say murine insulin. The insulin come from which animal? Marine? No. You almost get it. It's a mammal. So murine is what animal? Rodent. Actually, it's not rodent. Mouse. Okay, murine is mouse. Okay, the mu is from your mus musculus. Okay, avine. Bird. Yeah, that's where you get your avian flu. This you probably will not know. Porcine. Who is that? Porcine. Pig. Yeah. Canine. You probably will not know this already. Canine is not cat. Why you love cat so much? Yeah, dog. Remember, it's Canis lupus familiaris, so it's canine. Okay. Feline, then is your cat. Okay. Ovine, this one you will not know as well. But make a guess. Cattle? No. Cattle is bovine. This is cow. Sheep. Yes. Ovine is sheep. Because the name for sheep is actually Ovis aries. Okay. So you kind of have to know a few of these names so that when people tell you which protein you're talking about, you know what on earth you're talking about. Okay. So these are yeah, right. So all these are kind of useful names for you to remember. Okay. All right. Big insulin. Let us see if there's any interesting. Some of it you probably know, like for example, what is this animal? The name already tells you, right? This is number 3. What is this animal? Camel, correct. Ovine aries, that's your cow. Bostaurus, hopefully you know. Homo sapiens, by now, if you do not know, then do something about yourself. Xenopus, also very common. What is Xenopus? Octopus? Yes, nothing to do with octopus. Although it's amphibious, but it's nothing to do with octopus. Frog, correct. Okay, so let's see. So it's actually quite important to know some of these names. To tell you the truth, I do not know a lot about it. Sometimes I just have to guess and see what all of it is. So Daniel Rio already done that. This I have no idea. Half of them I don't know. Okay. Gorilla is just gorilla. The scientific name for gorilla is gorilla. Okay, so I will post up the tutorial questions Tutorial question is I would say tricky, but if you have managed to do it last week plus this week's Biopython, you should be able to handle a tutorial. Okay, so that's all for today. Have fun. Okay, bye bye. Anyway, I started marking some of your